name is Milka Koupenova. I am the chair of the Kenneth Brinkhous Award Committee, and I have with me one of the finalists, Kimberly Martinod, who is going to tell us a little bit about her research.
Thank you, Dr. Koupenova. It's really a pleasure to be here in Baltimore presenting my lab's work on Staphylococcus aureus sepsis. We are studying neutrophil extracellular traps, or NETs, which is a fascinating biological process where neutrophils release their extracellular DNA, which was originally described to be protective in terms of bacterial infection, but has since been shown to be potentially detrimental, especially when they're forming in the bloodstream. So we are looking now at the interaction between Staph aureus and neutrophils in the bloodstream in Staph aureus sepsis, and Staph aureus in particular because it is a really nasty pathogen, highly virulent, and also very prone to antibiotic resistance, so definitely a growing health concern. It is also a fascinating pathogen in that it can manipulate the coagulation cascade, manipulate platelets by interacting with them, and also evade immune responses from neutrophils, both in terms of net formation, where it can promote their formation, but also degrade them as well. So it really utilizes the host's own defense systems to its advantage. So we've been studying this in mice, which is rather difficult because mice are quite protected from these type of sepsis responses. So we've really optimized a mouse model to be able to study this in a way that we think better reflects the human disease. And we looked at the role for NETs using a genetic knockout of PAD4. And what we saw is that the mice that don't make NETs have no phenotype. So actually, they don't respond better or worse, which we were surprised to see. What we found, though, is when we boosted the neutrophil counts by giving mice G-CSF in advance, then they were strongly protected. And there, the NET deficient mice were doing slightly worse. So we wanted to dive into why that was. And what we found, and what has previously been reported, is that Staph aureus first goes to the liver, where it can be pretty rapidly cleared, but then once it gets to the kidney, it really stays there, persists. And neutrophils try to, let's say, fight it by forming abscesses. But again, the master manipulator is able to evade those. So we are, let's say, finding out that the more we study these, the less we know. What we thought would be a pretty obvious study turned out basically to be looking at this balance back and forth, bacteria, neutrophil, coagulation, and in the kidney in particular, it's important.
So tell me a little bit about the fact that these neutrophils that you find in the setting of Staph aureus are actually not thrombotic.
They can be prothrombotic to a degree. And actually, we think there is still a very important role for their thrombotic effect there in terms of kind of containing the pathogen. But what we found, what we were intrigued to find, is that the thrombosis was not necessarily pathological. So when we boosted the neutrophil counts, we had more neutrophils, more NETs, and we still had plenty of thrombi. So the thrombi are there, but the mice are still doing really well, and they survive their sepsis infection for a long time.
And so do you think at one point these studies can be extended to humans? And since the counts between neutrophils and platelets in mice and humans vary a little bit, that would be, something that we can consider in the future.
Yeah, absolutely. So we certainly see that there's a human equivalence of this, so we can identify high levels of NETs and also these immunothrombotic factors in patients as well. Targeting this, I think, let's say just targeting the NETs themselves, probably not enough. I think we need to target also the bacteria, probably the bacteria and the neutrophil interaction. There is some protective effect of NETs, but potentially some harmful effect as well.
Thank you for this interview, and good luck on your presentation, and thank you for joining us.
Thank you.